pleasure to have Ashutosh Srivastava. Ashutosh briefly did his PhD with Dr. Somdatta at CCMB Hyderabad. Subsequently, he had about 5 years of postdoctoral training in Japan. And for last 3 years, he has been a faculty at the IIT Gandhinagar. Also, he happens to be a collaborator of mine. Looking forward to Ashutosh your talk.
Thank you so much, Arijit, for inviting me, Mayappan, for organizing this conference. Looks for the last 1 day that I've been awake, I have been listening to so many exciting things, such a diverse lineup. So thank you so much for that. As he mentioned, I am currently working at IIT Gandhinagar for last 3 years. And I'll just tell you briefly about what we do. I am not going to ask you to remain awake because I myself am having very hard time remaining awake as of now. So feel free to go to sleep if that's what you want and that's what you like. That's completely fine, especially to the students sitting at the back. I'm not going to bother you. All right. So I come from IIT Gandhinagar, and just to give you a glimpse so that students get attracted and they come and join my group for different things. This is how it looks. We have a small group of PhD students, master students, computational biology group at IIT Gandhinagar. And the work that I'm going to present today is primarily done by Shreyansh. And Karthik. Shashank and Yashasvi are other PhD students. And if you get interested in what I'm talking about, you can reach out to them. You can reach out to me, talk to me more about it. All right. So, sorry for that. I was making these slides when I was half asleep, so sorry. So basically, these are 4 major areas in which our group works at IIT Gandhinagar. One major area that we work on is essentially modeling and dynamics of biomacromolecules. In this, we try to use integrative modeling methods, which essentially means we try to collate all possible experimental information which is there. It could be at different spatial resolution. It could be at different temporal resolution. We try to collate all of that, use computational methods to come up with the structure and also in some ways to study the dynamics of macromolecules. Some part of it I'll show today. The other area of interest is dynamics-driven drug discovery. Here, this is kind of routine where we look at certain proteins which are certain drug targets. And we try to understand their dynamics. We try to target them using small molecules and so on. This is a very new area, which is mathematical modeling. And just one student has started where we are trying to model plant circadian rhythms. I am also very new to this. So if you ask me questions, maybe I'll not be able to tell you the answers. The other area which we have a lot of interest in is network biology. Here we work at the level of protein structures, where we try to model protein structures as networks. This is some of the work that I did during my PhD. We are also looking to now expand these to protein-protein interaction networks using some of the recent formalisms in graph theory. So these are different aspects in which our group works. Now coming to the topic, which is circadian rhythms. Just to get an idea amongst the students, have you heard of circadian rhythms before? I see some of the heads nodding. That's good. So these are essentially day-long rhythms of physiological and behavioral changes which happen in almost all organisms. These are more apparent in higher organisms, but they have also been recently found in prokaryotes, for example. And these are a form of adaptation. So you can imagine that as the day goes by, there are so many environmental changes that happen from morning until night. And so all organisms have to adapt to these changes. So these are form of adaptation to changes in external cues. Now, what is also very interesting is that this is not just adaptation. That means it's not just responsive, but it is also predictive. So all biological systems, they are able to predict what the conditions are going to be and change accordingly. So this is another very important aspect of circadian rhythms. In case of mammals, this is governed by an area in the brain which is called suprachiasmatic nucleus. It's also called as master pacemaker, which regulates this clock centrally and taking primarily the cues of light through eyes. However, almost every cell in the body has its own mechanism, which I'll show in a moment, to to regulate these circadian clock or the rhythms that I'm talking about. Now, why are we trying to study it? Of course, it is interesting how it happens, but what has also been found is that the perturbation in these circadian clocks also are related to several different diseases. Most apparent ones, of course, have to do with sleep disorders, there are genetic disorders, as well as insomnia and so on. But there close connections have been found between the circadian regulation of the circadian rhythms and cancer, cardiovascular diseases, metabolic diseases. And those of you who have traveled or crossed time zones have already felt the adverse effects of perturbation in circadian rhythms in the form of what you refer to as jet lag. That is essentially nothing but a desynchronization of your central clock and your peripheral clock, which is the clock which is present in your body. All right, so how does it happen? Please don't get scared. Those of you who are not from biology background, who are not used to looking at such diagrams, even I also don't understand all of it. So I'll just refer you to some of the things which are important. So this entire mechanism, most of it which has been mapped in case of mammals, let's say I'm showing you for mammals, essentially contains transcription, translation, feedback loop. What it essentially means is that there are certain transcription factors, one of which I'm showing here, which lead to transcription of certain genes, which then go on to get translated, make certain proteins, make certain complexes, come back, stop their own transcription. Okay, so this creates a nice feedback loop. If this is getting too confusing, I'll now present a much simpler schematic to which I'll refer to during the rest of my talk. Okay, so it's the same thing. You have a couple of transcription factors which form heterodimers. Most of these transcription factors work as heterodimer. That means one type of one type of the other. They come together, form complex, bind to certain regions of DNA, lead to transcription of these genes. These are also referred to as clock controlled genes, which essentially means they are under the control of clock and lead to biological clock or in some way affect biological clock. Now these proteins, after getting translated, they form complexes, go back, stop their own transcription. So this is a nice feedback loop that exists. There is an accessory loop which is there, which I am showing here, where some other elements on the DNA, there are other transcription factors which I am showing here. These are referred to as ROR. You may forget the name, that's completely fine. These go and bind to this particular region, which leads to control of BMAL1. You may observe that this is here. So the control of this particular gene is under this particular loop. So there's a connected accessory loop which is there. I'll be referring to both of these in today's talk. First about this and then a little bit about this. What this negative feedback loop essentially leads to is this nice rhythmic expression of genes which have a period of approximately 24 hours, and that is why it is called circadian. Okay, so this is a schematic. This is the actual experimental data where people have found that there is this nice rhythms of expression of these genes, and there are many papers on this. All right. So accessory loop, I'll be talking about first of all, I don't understand why it is called an accessory loop, because it is extremely important. It should be a part of core loop, but people are calling it accessory loop, so I'm fine with that. So I'll be talking about this particular protein called Rev-Erb, okay, which is a negative controller here. So essentially it is a transcriptional repressor. It's also a nuclear receptor. So essentially there are certain ligands which bind to it. It goes into the nucleus. It goes and binds to this region and stops the transcription of these genes. Okay, so it's a negative regulator here of this particular loop, and this is the protein that we are interested in. Why we are interested in this? Because it's a active drug discovery target. There have been enough studies now which tell us that both upregulation as well as downregulation of this particular gene has issues. It creates issues. So both upregulation and downregulation has been implicated in a lot of different diseases. Now, I told you that this is a nuclear receptor. which essentially means that it can bind to either agonists. Agonists would be the molecules which will increase its activity or lead to its activity, which is the repression of the downstream genes here. Antagonists are going to be the molecules which will stop its activity and hence lead to expression of this. Okay, slightly confusing. So if you basically enhance the activity of this particular protein, there will be more repression. If you decrease the activity of this particular protein using antagonist, this expression will increase. Okay, I already told you that both of these have bad implications, and it has been found that, as you can see, that there is a role in Parkinson's disease, there is a role in muscular dystrophy, in type 2 diabetes. So people have been trying to find small molecules which can target these. Now, one of the issues with these small molecules, which have been found mostly by using FRET assays and chemical screening, is that you find a molecule which has an effect. The downstream effect is what you can see. The problem is sometimes they are agonists, sometimes they are antagonists, and while you are discovering it, you don't know what kind of molecules will come up. So certain antagonists have been discovered, certain agonists have been discovered. However, the structural mechanism of how this agonist works at the molecular level or how this antagonist works is not known. So this is essentially what we set out to do. As I told you, it's a member of nuclear receptor family. These are ligand-regulated transcription factors. Some of the common examples, if you have not heard of Rev, is vitamin D receptor, estrogen receptor, and so on. The canonical nuclear receptors look something like this. This is called as a domain architecture diagram, where I'm showing different domains. So it has an N-terminal domain, a DNA binding domain, a ligand binding domain. Okay, so the ligand comes and binds to this domain, and then this DNA binding domain goes and attaches to DNA and then does its function. All right. In case of Rev-Erb, which is a special class of nuclear receptors, this last part is missing. Now, what this last part does is that it enables these nuclear receptors to have both kinds of activities, activating as well as repressing. However, Rev-Erb becomes special that it can only repress. Okay, so that means it becomes an exclusive transcriptional repressor. It cannot activate. All right. What I'm showing you here is the ligand binding domain of this particular protein, and we'll be talking about just the ligand binding domain in the rest of the talk. Heme is the endogenous ligand for this particular protein. So all of you are aware of heme, which is also found in hemoglobin. This heme actually goes and binds to this particular pocket of ligand binding domain and acts as an agonist. So essentially heme is bound and then it can do more things. Essentially when heme binds, it enables this particular protein to bind to its co-repressors, other proteins, which when come and bind to this, lead to repression. Okay. So we wanted to understand what is the structural basis of antagonist and agonist binding in this particular ligand binding domain. For this, first, what we did is we looked at the conformational diversity of Rev-Erb across all the crystal structures which are present in PDB, which are not a lot. So there are about 8 or 9 structures present for this particular protein, not a lot of crystal structures in different states. So I'm just showing some of these here. So this, for example, is an apo structure, which essentially means there's no ligand bound to it. There's nothing bound to it. Okay. This is also an apo structure. This is a heme bound structure. This is a structure in which heme as well as a part of co-repressor, which it essentially brings in to do the transcription, is bound. Okay. This is another such structure in which other part of co-repressor is bound. And so what my student Shriyansh did, he took all of these structures and then just clustered them based on their RMSD. It's a very simple clustering which was done based on the RMSD of these different structures. And what you can very easily see that all the holo structures, which are essentially bound structures, they form a different cluster, and the apo structures form a different cluster, which essentially means that the binding pocket itself has enough conformational diversity in the apo and the bound state. Okay. Now, in the bound state also, if you see, the ones which are only bound to heme separate out, and the ones which are bound to heme as well as the interacting co-repressor, they separate out. Okay. So what we know from here is that there is enough conformational diversity that exists due to or at the binding pocket. So then he went on to perform long MD simulations of this particular pocket in different states. And I'll take you through these different states one by one. So the first simulation here that you see is of APO, nothing bound, no ligand, all right. And the second simulation that you see here is sort of a control or a comparative simulation in which what we wanted to do is to look at if you remove heme from the bound form, what happens to the structure, okay. So this is what he did. He took this structure in which heme was bound in the crystal structure, removed the heme from it, and then ran the simulation, okay. And what we could see, if you look here slightly carefully, is that this particular helix starts to go inside. See here? So it starts to go inside, and this is the binding pocket. This is where heme was there. And so he quantified it using these very simple distance measures where we are looking at the distance between different helices. This is the helix that you see here. And he quantified this by using these very simple distances between helix 3 and helix 11, helix 3 and helix 5. And as you can see, the distance between helix 3 and helix 11, they overlap completely, okay. So essentially, when you remove the heme, this helix completely goes inside and occupies the binding pocket, okay. All right. So then we wanted to understand why this is happening and what is the role of heme there. So he essentially then simulated this entire protein with heme in it, with heme in it. And what we found that when heme is present, then of course this helix does not come in, which essentially means that it is the heme which is, or the bound ligand which is holding this binding pocket in this open conformation. In the absence of the ligand, the pocket actually closes. We ran this simulation for a very long time just in the hopes of seeing that perhaps this helix will come out and there will be a conformational change which we'll be able to observe. However, we do not do that. So it seems like heme induces this kind of conformation in the binding pocket. The other thing that we could see is that this heme actually binds pretty stably there, which is not surprising. It has to be there. It makes these very stable contacts with certain histidine and glutamate here. And what we could see is that once it is there, the binding pocket becomes much more stable. There's not a lot of flexibility that exists in the binding pocket, okay. And if you look at the distances, I wanted to focus here. This is the heme bound form, okay. This is the heme bound form. This is the form in which we had removed the heme, and this is the APO form. So APO and this is almost same, whereas in case of 3CQV, these distances are higher. That means the pocket is larger, okay. All right. This is the movie of the same thing. I don't know why it's running. Okay. So the next thing that we wanted to understand, so we understand that the ligand goes there, stabilizes the binding pocket. Now, how does it affect the corepressor? Heme, as I said, is a ligand which is required for its activity. So if the heme is bound, it is trying to, then that means that the corepressor can be recruited, okay. So we took the structure in which both heme as well as the corepressor was bound, and we simulated that, and we made a slight variation of it in which we removed the heme. So essentially now you have one structure in which both heme and corepressor are there, and one structure in which heme has been removed and just the corepressor is there. And we wanted to compare that. And if you see these two simulations, which is essentially that, if you can see here, what we find is that when you remove the heme from here, okay, there is slightly higher. So you might not be able to see, but this is where that peptide is. This is the corepressor peptide right here, small one. This is more clear. So this is the corepressor peptide, which is here. And so if you look at this one here, when the heme is bound, this is much more stable. When the heme is not bound, your corepressor is actually moving quite a bit, which we have quantified here in terms of RMSF. In case of this ID1 here, essentially what happens is that this region you have higher flexibility in case of unbound, whereas this region you have lower flexibility in case of heme bound. So essentially what happens is that this region here interacts with the conserved region there and essentially stabilizes this. we further go on to quantify this, where what we have seen is that when an agonist is bound, okay, so, all right, so I told you that there is the heme, the binding of heme essentially leads to a recruitment of Corepressor. Agonist would do the same thing. Agonist would lead to higher binding of Corepressor. Antagonist will do the opposite thing. That means it will lead to lesser binding of Corepressor. So we then my student took all the antagonists and agonists which are known experimentally for this particular protein, and he docked all of these in this particular pocket, and we ran the simulation. So I'm just showing you result for one of the agonist and one of the antagonist. And what you could see is that in case of agonist, there are these interactions which are being formed with the conserved regions of Rev-Erb and the ID peptide, whereas in case of antagonist, we don't see these. In case of antagonist, if you can see the video, you can also appreciate that there is this movement of helix away from the pocket as this simulation proceeds. So this binding pocket just opens up completely here, and the interactions here are lost. Okay. When we further quantify it, we actually do see that there is indeed a difference in the interaction patterns in this particular region, which is known to be conserved, and there is a prior mutational study where the interaction between this region and the ID peptide or the Corepressor peptide has led to removal of this Corepressor or not binding of this Corepressor to Rev-Erb. Okay. We also quantified this based on simple RMSF measure between agonist, antagonist, heme, and non-ligand bound. And as you can see here, in case of agonist, in case of antagonist, you have very high RMSF, that means very high flexibility. Whereas in case of both heme as well as agonist, you don't have that much flexibility, and it's more like a no ligand bound. All right. There are other analysis that we did. I'm not presenting them here, but essentially what we found is that agonists and antagonists, they work differentially by affecting the binding of Corepressor. We have been able to identify the region which is responsible for this. So binding of ligand, what is it doing to particular regions of the helices of Corepressor and Rev-Erb, which lead to this particular effect. Agonists, including heme, stabilize the binding of Corepressor by increasing the interactions with conserved regions of Corepressor and ligand binding domain. Antagonists perturb these and lead to lower Corepressor engagement. So this is how basically we have been able to identify. Now the next step, of course, becomes that we are now trying to see if we can rationally develop better agonists and antagonists against this particular molecule. Okay. So this is one part of the work. By the way, all of this is unpublished. The first part we are going to soon we are going to communicate. This is the other story which is still developing, where I'm going to talk about this core loop, as it is called. And as I told you, there is this heterodimer which is present of clock BMAL. It binds to this region and leads to transcription, which is then stopped. Okay. So if we look at the domain architecture of this particular transcription factors, these belong to a class of transcription factors called BHLH, pass domain transcription factors. These are again ubiquitously found in almost all animals, and they do a lot of different work, different effects, including hypoxia-inducible factor, for example, which is very common and a target for cancer. So these particular transcription factors, their heterodimer structure for human was actually not known. For mouse, it was known. So my student, he modeled this, predicted it using AlphaFold, which is very easy now. Then he merged this with the DNA bound model. We got this, validated this model, and ultimately he performed long MD simulation. These are essentially 5 1 microsecond simulations of this entire system of DNA bound, enhancer box bound transcription factor. And what you can immediately see here is this nice domain motion which is happening from the DNA bound region, which is shown here, and the pass domains, which I'm showing here. Okay. Now, the very first time when we saw this, we were like, okay, it may be functional, it may not be functional, it might just be an artifact of the simulation. And so we started looking at all the transcription factors and their structures. We found that actually this kind of motion could be possible. However, we did not observe such conformational diversity in these different transcription factors. All right, I'll come to that a little later. So we tried to quantify this large-scale domain motions. If you look at the domain-wise RMSDs, what we found is that the BHLH domain and the PAS-A domain, which is here, shows slightly higher flexibility as compared to the PAS-B domain. That is also understandable because it is a PAS-B domain which forms the largest interface between these two heterodimers and might be stable because of that. We then quantified the overall directions of these motion. This can be done very easily using principal components analysis of this entire trajectory. And this is a simple coordinate-based principal component analysis in which you can then see the directions of these large motions. And there was this bending motion which was there and the twisting motion which was also visible in the trajectories. So we went ahead, we clustered the PC1, PC2 space. Just the PC1, PC2 space explains around 60% of the overall variance in this total domain motions. And when you cluster this, what you can see are the different conformations which are populated in this entire space. And what you can also see is that the distance between the two domains, the motion which was happening, is different at these different locations. Okay, it can go as low as 61.6 angstrom here to as large as 76.2. So there's almost a motion of 15 angstrom or so, 14 to 15 angstrom, which happens in this particular case. All right. So while we were looking at it, Karthik, who's my student who's doing this work, realized that there was actually a pocket right here. He was looking for pockets, so he found a pocket. Okay, so right at this hinge region. Initially, we didn't think much about it, but then when we started thinking more about it and looked at it, we found that actually the size of this pocket increases as the distance between the two domains increase. So while it is functional and it is undergoing this motion, there's a possibility that you can target this hinge region here and affect its function. So that was his interest. He wanted to pursue it. So we are currently trying to see if we can target this particular pocket, which would then have effect on these motions. Okay. All right. Yeah, so the pocket is somewhere here at in this particular region. Okay. The other interesting phenomena in this particular in this entire complex is that there are 4 disordered loops. If you combine both the heterodimers, there are 4 disordered loops, some small, some long. We are currently trying to map the entire conformational landscape of these 4 loops, and the reason is that this loop here and this loop here, the conformation of these loops affect the motion of these domains. So in what kind of conformation these loops are ultimately affects what kind of domain motions this entire heterodimer shows. All right. So as I said, this is still an ongoing work. We are still analyzing these trajectories more. He's also trying to understand if there are certain phosphorylation sites here and they are known to affect the repression. So we are trying to understand how that happens. So there are large functional domain motions observed in clock BMAL1 transcription factor. Large, you understand, you have already seen about 15. Why is it functional? Why am I calling it functional? Why is it relevant? In 2023, this paper came of one of my previous collaborators, who actually solved the structure, the cryo-EM structure of this particular transcription factor bound to the nucleosome. Now, if you look at, this is the density for, it's not a great cryo-EM structure. The density is not, the resolution is not that great. They had to actually cut the heterodimer, the 2 domains, the large domains, into 2 different pieces and then fit it here. Essentially because based on the crystal structure, there is absolutely no way that you can think of this particular conformation. It's a very bent, weird conformation of the PAS domains here and the BHLH domain. And see this beautifully binding to DNA here when it is opening up the nucleosome right here. So that's when we realize that the kind of domain motions which we are seeing are indeed functional. They are possible. And in fact, they are required for this transcription factor to work. Okay. And so we are now so a student is now trying to actually use these trajectories to essentially find out the conformation in which this is present. Slightly hard because this could be an induced conformation. but we are looking to find to essentially perturb the structure so that it can bind this and we can get this particular conformation. There's no atomic resolution structure of any transcription factor, BHLS domain transcription factor, which is anywhere close to this. All right. So here I'll end my talk. All the work was done by Shriyansh and Karthik with some work from Rakesh, who was a master's student. I would like to thank all the collaborators within IITGN, out of IITGN, Arijit, who has been talking to me, guiding me, helping me for a long time, Madhulika, with whom I'm doing a project currently, Sarvanan, Shravan, and other collaborators, funding agencies for their generous support for the work that we do. All right, thank you so much.
We have time for a few questions.
Had a high-level question on, like you mentioned the nuclear receptors have
Sorry?
in the first part of the talk, you talked about different antagonists and agonists,
Yes.
right? Like, how does the nuclear receptor differentiate between different agonists or antagonists, same kind?
Yes. Most of the times these are context dependent. So essentially for a particular kind of signaling, a particular kind of ligand will come and bind and then lead to the signaling. In this case, we don't know. So essentially, as I said, these molecules were developed based on very simple FRET assays, which essentially just quantify that if the ligand goes and this goes and binds to DNA, what happens in the downstream genes. Okay. So when they found, they were not actually looking for agonist or an antagonist. It's the downstream effect which defined, okay, this is an agonist and this is an antagonist. Structural level, no information how it works or how the protein selects.
Yeah, so just an extension to this question, it would be great if you can simulate with the 3-4 agonist and 3-4 antagonist so that you will get a better insight.
Yes.
Just a suggestion here, you can also calculate the angle between helices, how it is changing when agonist binds and antagonist bind. There is a twist kind of thing also maybe there in the involvement of other residues. Like you have already done for one, but if you get for multiple ligands,
Correct.
Can give you a better insight.
Correct. We have already done that. I did not have time to present all of this. So this work is almost ready. He took 5 agonists, 5 antagonists, did all the simulations. We see some consistent result. There's slight complexity in this. 2021, there was a Science Advances paper which talked about binding of heme, which essentially proved that heme constitutively binds to this and recruits the corepressor. What they found is that the effect of binding of heme is not uniform on the 2 peptides that I am showing, ID1 peptide, ID2. So these are 2 parts of a large corepressor which come and bind to this and then essentially stop HDACs and so on and so forth. Now, what they found using their ITC studies is that when the heme binds, ideally you should have more interaction with both. That doesn't happen. So there's more interaction with one, there's less interaction with the other. Overall effect is that it is more stable. We see something similar where we don't see the uniform. So there's even the effect of agonist binding to the pocket is differential on the 2 peptides that we see. But yes, you're right. We have looked at other agonists and antagonists. We have not simulated all of them. I think we have simulated top 2 or top 3 agonists and antagonists. And these are not randomly selected. These are known, experimentally proven, experimentally known agonists and antagonists.
Fantastic.
I have several questions. Maybe I'll talk to you later. One of the main things is this: is other crystal structures or NMR structures of the agonist and antagonist binding this system, and how do they displace the heme? Because heme would bind, so your binding energy for the heme would be much more than these.
No crystal structures. Let me tell you this. Okay, while we were working on it, there is a isoform of this particular protein. So that is called Rev-Erb alpha. there's Rev-Erb alpha and beta. We are working on beta. Alpha is very similar. I think sequence identity of about 60% or so. They're very similar. Functionally, it's very difficult to differentiate which one is working, whether it's alpha or beta. Most of these molecules have been developed for alpha. Okay. So while we were working, 2023 end, in fact, a paper came which describes the structure of alpha, with the first ever structure of alpha with a synthetic ligand. Up until now, the only structures which are available are with heme. No structures with synthetic, neither NMR nor crystal structures with any of the synthetic molecules. And that is why it has been so challenging to understand how these agonists and antagonists work. Yes, I know. It's going to be very hard to displace the heme, but these seem to do that, because that's why we did not go the route of finding it randomly, because we don't know then. These are experimentally proven, and they, by ITC studies, they do show that they actually displace the heme. Now, how that happens, we don't know, because this is, in all likelihood, this is an induced fit mechanism of binding.